All right, this is going to be 2 videos over the muscular system. This is the first one, and the second one is located below this one. The muscular system is one of the more complicated body systems you can cover. In fact, from this point on in your anatomy and physiology studies, we are only covering organ systems. We did that with the skeletal system. It started with the integumentary system, but these are more detailed and will continue that way throughout the rest of anatomy and physiology when you take the second half. So a couple of things about muscles first. Remember that a property of muscle is the ability to contract, to shorten and develop tension. That's actually called the sliding filament theory. We'll get to that in the next video. Production of heat is a byproduct of muscles contracting. That's why when a person's cold or they're running a fever, they shiver. Involuntary contraction of very small muscles generates heat. And there are different functions for muscles. This section we are going to mainly cover skeletal. In fact, that's all we're going to talk about. The whole process of how they contract is very similar between all of them, but as you know, muscle has different properties. There are 3 types of muscle. This is skeletal, this is cardiac, you can see the intercalated discs, and this is smooth. This section we are going to deal with skeletal muscle. So just starting here first, just some terminology that we're going to go through. Anyway, fascia, tendons, and aponeuroses. You do have I got a double there. You have a PowerPoint that comes with this section. You can see here an aponeurosis. There's one on top of the head, because this is actually called the frontalis muscle. There's a muscle on the back called the occipitalis that aponeurosis connects the two together. There's also one down here in the abdomen. It's a sheet of connective tissue that connects a group of muscle or muscles together. You remember tendons connect muscle to bone, and then ligaments connect bones to bones. But fascia, and we'll get to this, fascia is something. Okay, so this person here, I'm sorry to balance back like that. If you were to take off, well, we used to dissect cats, and you spent the first 2 days of lab trying to get all the fascia off. You can see on this muscle model, you can actually see the skeletal muscles. That's because the fascia has been taken off. And see it there, that white layer? Now it covers all the muscles, and then underneath the fascia you would have layers of muscle. So that would be the next part, but fascia is a layer of connective tissue that covers all the muscles externally, like it says there. A tendon connects muscle to bone. We talked about that. And say, I don't mean to bounce again, there's a tendon right there, you can see that. What bone is that? Let's see, 3, 2, 1. Yes, that's a femur. But see the tendons connecting, and then you have fascia, and then you have all this other part here. Now, on your page, muscle breakdown, well, and it's probably, let me be a little further along here. I like that. That's a really good diagram. Okay, so, where I was going is that's in your PowerPoint as well. So you see how this is all broken down. So you can take a muscle like this. This is like a quadricep. And you see how you can break it down to microfilaments. That's what we're going to do. And it's doing it there. Muscle, fascicles, muscle fibers, myofibrils, thick and thin filaments. And that's the level we will stop as far as detail goes. So but I'm going to stay with our outline here, and let me see if I can make this a little more centered. There we go. All right, so we're going to start our journey here, and I'm going to go up to the point where we go through the whole thing together. So muscles are highly organized. These are the layers. In fact, the whole body is made of layers, but we're talking about muscle here. Muscle also has layers. The epimysium is the layer. Now, fascia is a layer of connective tissue that covers everything. Now we're talking about individual muscles, but epimysium would be this outer layer. See that white layer, or the clear, looks like saran wrap, and then there's a tendon. So the fascia is gone, So that is epimysium, that layer on top right there. Covers all of the muscles externally. Perimysium. Now what happens, this whole thing is a muscle, but you'll notice that this muscle is separated into compartments. If you pull one of those out, and it's called a fascicle, what that is showing is an individual muscle fiber that's being pulled out there. But a fascicle is a bundle of fibers, and you can see there's 1, 2, 3, 4, there's 5 muscle fibers there. If you're wondering what is that, that's a muscle fiber. So there's 8 fascicles there, and they all have individual, because muscles, that allows you to have control. You're not always going to use every ounce of muscle within a muscle. All your movements would be huge and jerky movements. There's times where you want to have fine motor control, and there's times where you want to be able to recruit more muscle fibers. So a fascicle as a compartment, it's a bundle of muscle fibers, and basically that white layer that surrounds it is called perimysium. And then if you pull it out like this, that's an individual muscle fiber. Now remember, muscle fibers are muscle cells, but they're called fibers because they're so long. There's a layer called endomysium that surrounds each individual muscle fiber. So muscles are highly organized. They have 3 layers to them: the epimysium, the perimysium, and the endomysium. Okay, a little bit of histology. The sarcolemma, and I believe it's probably on the next page. No. Interesting. That's all right. Yeah, so histology. Sarcolemma, that is the cell membrane of a muscle fiber. And my brain may be leaving me. I swear that's supposed to be there, but oh well. So here is, well, I have this up. So this is one of those, this is a muscle fiber. See the nucleus, and remember skeletal muscle has multiple nuclei, it's multinucleated. If you go through the histology or the cell level of a muscle fiber, you have these little cylinders here. Those are called myofibrils. You have this here on the outside. You see it's called the sarcolemma. It's polarized. Muscle cells have a positive negative charge across the membrane, so it allows them to react to a stimulus. You have this inside part. This is called the Sarcoplasmic Reticulum. And then you have, these are transverse tubules, these little parts right in the middle, and we'll get to that. There's some mitochondria, and then like I said, you have the myofibrils, which are made of myofilaments, and then you have the cisternae of the sarcoplasmic reticulum on each side, and we'll get to all that. So the sarcolemma would be the cell membrane. It is polarized, and in other words, it looks like this. That's the resting membrane potential of a sarcolemma. See the little plus on the outside, minus on the inside? It means it's polarized. That's how muscle fibers react to an electrical stimulus. They have to be polarized. There's more sodium ions, the Na+, located on the outside of muscle cells in any cell in your body. There's more potassium ions, the K+, on the inside. So that's a natural setup. Sarcoplasm would be like cytoplasm, what all this is sitting in, a jelly material. Myofibrils, which are made of myofilaments, which are thick and thin filaments. These little cylinders here, and there's 1, 2, 3, 4. There's 9 there. There's 2 more on top. So there's 11. Those are myofibrils. And you can actually see there's a couple of dots. There's a red dot and a little smaller blue dot. Those are the thick and thin filaments that make up each myofibril. If you get motivated and you decide you want to lift weights and build strength, you actually add more to these little cylinders here. That's how muscles increase in size. Now, a sarcomere. And if we were in a face-to-face class, we would draw it right here, but that's okay, so we have a diagram right here that shows one. Sarcomeres are little areas along a myofibril, and you can see them in that picture. Do you see the alternating colors along this? And you can see a line here and a line right there. These are called Z lines. A sarcomere is between each one of those lines. So they're like little box cars. So a myofibril is basically a whole bunch of sarcomeres end to end along a muscle fiber. Yeah, it is the contractile unit. Like I said, it's called the sliding filament theory. And we'll get to that. But that part shortens. And then the whole muscle fiber, the myofibril shortens, which leads to the entire muscle fiber shortening. Okay, parts of a sarcomere. I-bands contain thin filaments, but no thick filaments. Extends from an A-band to the next A-band. Now, I-bands are kind of lighter colored. So here's the first thing. This little thing that goes down here, this part, that's called a Z line. See that? It's the boundary between adjacent sarcomeres. That lighter color area is an A-band. I thought I might have it on here, but I don't. The darker area on each side here are the A-bands. So this is the I-band. I hope I said I-band a while ago, the lighter area. A-bands, this is dense regions that contain thick filaments, and you can see them there, the red. These are the A-bands. This is actually something called an M line that goes right down the middle. And then, like I said, these are two Z lines. Now, on here, we're going to have a labeled sarcomere somewhere, I would think. Well, here is, so this picture I was showing you, this is a myofibril that's been pulled out. And you can see the M line there. And you can see the red is the thick filament. See that? The blue are the thin filaments. And then there's the Z line. Here we go. Let's blow this up. So A-band, like I was saying, the darker colored here. I-bands are there. And then the Z line is basically the distance between, so here to here is one sarcomere. And then from here to there is another sarcomere. So the I-bands are the lighter areas. These are the A-bands. So there's an I-band on that side, an I-band on that side, or just in here between. Okay, basically a Z line goes right down the middle of an I-band. How's that? Here's the A-band. Here's something called the M line, a little thing of proteins, and the H zone would be the lighter area on each side of an M line. Basically, when a muscle contracts, these red thick filaments, they stay in the same place and they pull the blue thin filaments toward the center, and that results in the muscle shortening overall. All right, so if you want, I'm trying to do this where you don't have to print anything out, of course, but so like I was saying, here's the Z line, I-band, this is the A-band, here's the M line, the H zone, but the A-bands are the dense on each side. And you can kind of see them there. See, the red would be the thick filaments, and this looks like it's purple. Those are the thin filaments. So if you blow those up under electron microscope, so we're talking about this red line right here now. If you get even more detailed, this is what a thick filament looks like. It's made of a protein called myosin, which is what that is. And then these little things here look like little golf club heads. Those are the myosin heads. And they're an integral part of when a muscle contracts. Just remember, when a muscle contracts, these little proteins, the red stays where it is. The little purple, bluish lines right there actually slide past the red and allows the entire muscle to shorten. So this is the myosin head on the end. Now the thin filaments, which are the little blue here, and even in my other, see the bluish to purple colored, not the red. The red's a thick filament. Thin filaments have 3 proteins, and it looks like this if you blow them up really big. The purple with that little dot right there is actin. It makes it the majority of a thin filament. And that little area right there is a binding site for the myosin heads. And when they stick there, it's called a cross bridge. The little head is actually designed to bind right here. When this is exposed, and you can see here it's covered up. See the little hole's actually covered up. When that part is uncovered, those little myosin heads, they reach up, they grab, and they pull. This blue long protein that covers up this binding site when a muscle is relaxed is called tropomyosin. Long molecule that covers the binding site on actin and prevents the actin-myosin interaction in a resting muscle fiber. And the yellow is a protein called troponin. And why is that important? Troponin binds to calcium ions, Ca++. Very important part of a muscle contraction. When a muscle depolarizes, in other words, when it's going to contract, it's been stimulated, calcium ions get released into the sarcoplasm and they bind to troponin. Troponin pulls over this little blue line, tropomyosin, exposing this binding site, and that little head reaches up, grabs it, and pulls. So just remember in our setup, see the red is right here, it stays where it is. It's going to reach up, grab, and pull these little blue lines closer to the center. And so when that's happening down the entire length of the myofibril, so this is shortening, that's shortening, that's shortening, it results in the entire muscle fiber shortening, which would then lead to the entire muscle shortening. So that's how a muscle contracts. It's called the sliding filament theory. And we have a few things left before we get there, and we'll start that in the next video. But the sarcoplasmic reticulum is the, see the blue? I like this picture better. So here are the myofibrils. You can see the areas, the sarcomere. Here's the two Z lines, but this whole thing is called a myofibril. And that little area between here and here is a sarcomere. But this blue is called the sarcoplasmic reticulum. See that? And then that tan tube that goes down the middle is referred to as a transverse tubule. Now, it actually connects to the sarcolemma, and that's how an electrical impulse called an action potential, that's how it gets deep inside of a muscle fiber or through those little tubes. So that's important. And the terminal cisternae is part of the sarcoplasmic reticulum. It's that area on each side of the transverse tubule. So here, let me blow this up bigger. Cisternae, see, all of this is the sarcoplasmic reticulum. It looks like a little sweater, including these thicker, whatever color that is, orangish tubes on each side. All that's part of the sarcoplasmic reticulum. That part is called the cisternae. It actually stores calcium ions. And the transverse tubule, you can see them there. They are continuous. If I go back up, see the openings here that are going into the, so this is the outside of a muscle fiber. See how they're going down that way? That's how an electrical impulse from the brain or another source gets deep inside of a muscle fiber. So that's the anatomy. Now, and like I said, here's the resting membrane. Okay, so let's go on to this is the complicated part of this. I'm going to go through this, and then the next video we're going to start and go through the whole thing. So, starting here first. A skeletal muscle contraction is the interaction of a cellular and chemical component. It's called a coupling. The chemical part has to do with neurotransmitters, and the cellular part is all the parts we just went through. Okay, a resting membrane. So now we're talking about a muscle fiber that's in a resting state. It's not contracting. On your PowerPoint that I gave you, this is the resting membrane potential of a muscle. The outside has now this, just pretend this area is a sarcolemma. The outside has a positive charge. The inside has a negative charge. There is more sodium ions on the outside of a muscle fiber, and there's more potassium ions on the inside of a muscle fiber. Now, there's something called the sodium-potassium pump. It's a continuous channel in every cell. Cells are always pumping sodium on the outside and always pumping potassium on the inside. So what you have here is a gradient. There's a ton of sodium on the outside. There's less sodium ions on the inside of a muscle fiber. There's a ton of potassium ions on the inside of a muscle fiber, and there's less on the outside. Now, you remember how diffusion works. Anytime you have a concentration gradient, a high concentration to low concentration, particles are always trying to move to the lower concentration. So sodium ions are trying to get inside here, and potassium are trying to leave. But they can't because they're not permeable to a cell membrane. So basically, they have to wait for something to happen to open a little door in the cell, the sarcolemma, so they can rush inside of a muscle fiber. Okay, here we go. Now we're going to start here first. Now, before I get to this, you have this picture, and I did provide for you a couple of diagrams. So here is, this is on your exam page. There's a neuromuscular junction. I believe that's the entire thing. That's actually pretty cool. I'm going to leave that one, and then I'll do this one on the next video. So, going here. Here's the spinal cord. This is actually an area called the dorsal root. This is a dorsal root ganglion, and I like doing it like this because you can click, see the plus, you can click and make it bigger. So here's the spinal cord. These are supposed to be the nerves coming off the spinal cord, and here they're innervating. You can see the individual muscle fibers. Here's the nuclei, and you see how they're spreading out like this, and these are the muscle fibers, and see here are the nerves that are innervating the muscle fibers. So there's a muscle, see it's pulled out, and it's showing all the nerve innervation attached to these muscle fibers. Now, if you blow up one of those little areas right in here, if you look at it, that's called a neuromuscular junction, and it looks like that. So one of those little extensions on the end here, if you blow that up huge, this is what it looks like. So here would be the muscle fiber, here's the sarcolemma, and here's the end of one of those nerve endings. Sometimes they're called knobs. This is axon terminal of neuromuscular junction. But you see here's the nerve coming down. These are mitochondria. And then this is all of the area what's called the neuromuscular junction. Now what happens is a nerve impulse, either as a reflex or from the brain, it comes down the spinal cord. It travels out this way. And you can see, now these are the sensory ones going in that. Well, anyway, these are not. These are both motor neurons. But see where they're connecting? So that's how a nerve impulse, an electrical impulse, is called an action potential. It travels down the spinal cord. It travels out into peripheral nerves. Anybody know the big nerve in the back of the leg, the hip area, the sciatic? It's a huge nerve. So you'd have little branches off the sciatic, and then would go down and innervate the lower leg muscles and hips, like that. And it would look like this. And when an electrical impulse reaches the end here, and you can see, look, a nerve actually does not attach to a muscle. It doesn't attach to anything. It doesn't attach to glands or organs. There's an area here called the synaptic cleft. And when an action potential reaches the end of one of these nerves, it causes the release of a neurotransmitter that crosses this little space and will go down here and bind to receptors in the muscle fiber. And at that point, the nerve is stimulated. And that's called depolarization. You see it there? A wave of depolarization? All right. So the next part to this, when a muscle depolarizes upon chemical, electrical, or mechanical stimulation, a muscle cell or nerve cell, the membrane suddenly becomes permeable to sodium ions. And what happens is sodium ions will start rushing, and it's not on here. Go ahead. Go away. Thank you. You can see it on this diagram. Sodium ions will start rushing across the muscle fiber, and it's carrying all these positive charges along with it because it's Na+. Sodium ions are cations, all those flushes rushing inside a muscle fiber will temporarily cause the membrane to switch. Remember, a resting membrane, the outside is positive. I guess I can go back to our picture. Remember, the resting membrane, a normal resting membrane, the outside positive, the inside's negative. When a muscle depolarizes, that charge switches temporarily across the membrane. The outside becomes negative and the inside becomes positive. And the reason that is, there's two things that happen. Sodium ions rush inside of a cell, causing a reversal of the electrical charges. The outer membrane becomes negative and the inner membrane becomes positive. That's depolarization. Nerves depolarize, muscles depolarize. On an EKG, when we study the P wave, the QRS, the T waves, a P wave is atrial depolarization. A QRS is ventricular depolarization. It's talking about the skeletal muscles, the cardiac muscles depolarizing. That's what's going on. So the entire fiber depolarizes, but it can't stay that way. Okay, the outside cannot stay negative and the inside positive. That's not the natural setup, and that's not a polarized membrane. So what happens right behind it, after a muscle depolarizes, and this is showing the plus moving in, you see that, okay? Right behind it, very quickly, it is repolarizing. And how does it do that? Well, when a muscle repolarizes, the little doors, the permeability for sodium stops, and the permeability for potassium ions increases, and potassium ions start flying out of the cell. They also have a positive charge. And so much flies out that it ends up restoring the resting membrane potential. It turns the outside of the cell positive compared to the inside, which is negative. Even though potassium has a positive charge, if you stack more positives on the outside of something compared to the inside, you're going to have a difference across the membrane, the outside being more positive, the inside being less positive. So when a muscle repolarizes, it's trying to restore the resting membrane potential. So during repolarization, potassium ions rush out of the muscle fiber, and like it says here, it occurs very rapidly. It's actually by diffusion, but will eventually return sodium to the outside, potassium back to the inner membrane, but this restores the original electrical charge of the outside being positive and negative on the inside. So it will return it back to that state right there. And that happens every time a muscle contracts. It will depolarize like this, and then right behind it, okay, there we go. I didn't mean to skip this. There's potassium moving out, and there it's showing the charge being restored. So that area depolarizing, and it's like a wave. It just travels down the entire muscle fiber. As this part is depolarizing, right behind it, it's repolarizing. And it has to do that. If it never repolarizes, it can't conduct another signal. And unfortunately, that's what asystole is. When if a person's heart stops, their heart depolarized, the cardiac muscles depolarized, and never repolarized again. Here's another picture showing how the little nerves attach to the muscle fibers, and then here's another picture of a neuromuscular junction. And there's the blue dots would be the neurotransmitter being released. So let's move down here. So here's a neuromuscular junction right there, and you can see the vesicles that are storing that. And like I said, I gave you a picture like this. The green dots, now the other one, it's I think they're blue, but these are vesicles. Now the neurotransmitter with skeletal muscle is called acetylcholine, and I would hope, where is it? Okay, there's the enzyme. There it is, acetylcholine. It's actually abbreviated as ACH. Acetylcholinesterase is the enzyme that breaks down acetylcholine. But at these nerve endings, acetylcholine, here are the vesicles. Now here's what happens. When a nerve impulse reaches the end of the nerve, these little vesicles, they move down here to the bottom, and they release this neurotransmitter into this space called the synaptic cleft. Now this area that's folded here is called the motor end plate. It's the folded sarcolemma. And there are the little channels. You can see those. The sodium-potassium channels, you can see those here. That's a good, you know, how sodium rushes inside, this little channel will open. That's why it's said to be more permeable. Here's a potassium channel that will open and allow potassium to leave as this one closes. But those little neurotransmitters, they'll come down here, they'll bind to these areas here, and then that will depolarize this area, and it will start that wave of depolarization. So it will start essentially that. See how it's a wave going this way, and then right behind it, it would repolarize again. So, and calcium ions are involved in this part. We don't talk about it much, but when they move in, that's the stimulus to release acetylcholine. Now, the minute that acetylcholine binds to these receptors, it gets broken down. It can't stay bound there, or the muscle would never relax. And that's what, there's an enzyme called acetylcholinesterase, great Scrabble word, ASE. It breaks down acetylcholine the instant that it binds to the little receptors in the neuromuscular junction. So, anyway, yeah, I know that's pretty complicated. We're going to start with the next video right here.